For today is Nir Yosef, who's an assistant professor in the Department of Electrical Engineering and Computer Science in the Center for Computational Biology at the University of California at Berkeley. He started there in January of 2014. Prior to that, he got his BS in Computer Science from Ben Gurion University, PhD in Computer Science from Tel Aviv University, and then he did postdocs at Harvard Medical Institute in the Harvard Medical School in the Broad Institute. His research interests are in developing computational methods for analyzing diverse high-throughput data sets with the goal of better understanding gene regulation in mammalian cells, and also dynamic regulatory circuits, focusing on the immune system as a model, which is why we were particularly excited that he was able to join us for this particular summer school. The title of his presentation is Reconstructing the TH17 Differentiation Network from Profiles to Drug Targets. Nir?
Right, good morning everybody. Can you hear me okay? Yes. Right, so thank you very much for the invitation. I'm really happy to be here and talk about some of the work that we've been doing the past couple of years. So I'm going to start with the motivational slide. So I guess I don't have to say this to you, since you are here and you care about creating models and stuff, but as a general, we see that high-throughput profiling methods are doing exceedingly well in identifying individual components that are important for the various systems that we study. It can be differentially methylated regions in the DNA, it can be differentially expressed genes or whatever. So we are good in identifying these specific components. But what we don't know, and often in order to understand the function of these components, we need to understand how do they come together as a system, how they interact with each other and perform some computation, some function that we care about. But this poses some great challenges. And the first challenge is how to design experiments, and then how to use these experiments to build these models that connect our components to each other. And in this example, and throughout my talk, except for one case, the networks that I will talk about connect transcriptional regulators to their target genes. So the second challenge, which is even harder, is how to extract from these networks that can often be very, very large, viable hypotheses that we actually want to go and to experiment in the test later. So basically, the challenge is to understand the function of these networks. And last thing, to make things even more complicated, these kind of networks, especially transcriptional regulatory networks, are far from static. They can change. They change between organisms, they change between tissues, they change between cell type, and even within the same cell type, they can rewire over time, and they can even be different between individual cells out of a seemingly homogeneous population of cells. So my lab at Berkeley is focused on this type of analysis, and specifically what we are interested in seeing is how can we understand the variation over time, the variation between individual cells, and how can we use this variation in order to say something about how a system is being regulated and what is the mechanism behind some phenomena that we see. And in order to illustrate this kind of work, I'm going to tell you about the work that we did in the past 4 years. We focused on a very specific system of T cells. So this is how our story begins. So when our immune system is being challenged by, let's say, by sensing some pathogen, one of the forms of the adaptive immune response is the activation of naive T cells. So when naive T cells become engaged, it can differentiate into one of several cell types, where the decision of the differentiation depends on the specific cytokines that are present in the microenvironment of the cell. Now, there are two major arms to this process. One is to, and this is like in a very, very crude terms. So one is to T cells that promote the immune response, and the other one is to T cells that suppress the immune response and promote tolerance. And as you can imagine, the balance between these two arms is very important, and any disturbance of this balance can have detrimental clinical effect, like various types of autoimmunities or immunodeficiencies. So the most recently discovered subset of T cells in the immunity arm is called TH17 cells. So TH17 cells were discovered not too long ago. They are recently, relatively recently discovered a subset of cells. I think it's like 2006. And since their discovery, they've attracted a lot of attention from the research community because there is an increasing body of evidence that shows that these cells are important for a large number of autoimmune diseases, and this is just a short list here, including multiple sclerosis, psoriasis, various types of IBD, and others. And so understanding the process by which a naive T cell can actually differentiate into Th17 cells, it's a very important question to us. It has a very, I guess, important translational potential to that question, to understand the mechanism here. So what do we know about Th17 differentiation? Well, I guess what did we know when we started this project? So first of all, and importantly, the differentiation process can be induced in vitro. We already know some of the major factors that transition factors that are important in this process, like STAT3 and IRF4 and BATF, that active pioneer factors, and gamma T, which is a specific to Th17 cells, and it's like a later induced factor in this process. And finally, in order for these cells to remain stable, the stability depends on signaling through IL-23. As for mechanism behind the balance between inflammation and tolerance, so we know two things. One is that there is a mutual antagonism between RR gamma T, which is a master regulator of Th17 cells, and FOXP3, which is the master regulators of the regulatory T cells. And this is one fact. The second fact is that Th17 cells, under some context, they can produce immunosuppressive cytokines like IL-10 and be less pathogenic. But despite these anecdotal, facts that we know about Th17 cells, there is still very partial knowledge about the mechanism behind Th17 differentiation. So what we set up to do in this project is to basically, we have this big goal in our head, which is to create a model of the transcriptional regulatory network that controls Th17 differentiation. And what does that mean? So we start with mechanistic questions, very basic mechanistic questions, like who are the key players in this process? Who do they interact with, and when do these interactions actually occur? And maybe most importantly, which perturbations can we do in order to disturb the differentiation process? And with these mechanistic questions answered, or at least we have hypotheses for them, we can start thinking about higher level functional questions, like how is the balance maintained between inflammation and tolerance? What's the mechanism? Or how is the pathogenic potential of Th17 cells is regulated, for example, through IL-10 production? Or what makes these cells stable? And then with these functional questions answered, we can start thinking about applications and see if the mechanisms that we identify can highlight potential good drug targets. So we have 4 stories that we kind of walked our way through these questions, and I'm going to tell you about 3 of them today. So basically just to walk you through these things that we did. So first we build a large network model of the regulatory network that controls Th17 differentiation over time. And then we focus on the question of stability and try to build a model of the signaling regulatory network or pathway that is actually downstream of IL-23. And then we have this collaboration with a pharmaceutical company where we tested the effects of drugs, specific drugs against RR gamma T, and see the effect of these different molecules on the network of Th17 differentiation. And it was interesting because actually different molecules have different effect on the network, even though all of them in the end lead to decrease in inflammation. And finally, I'm going to talk about how we can tease out different subtypes of Th17 cells and see if we can actually isolate the pathogenic Th17 cells from the non-pathogenic one. And this is done using single-cell RNA sequencing. So I'm going to tell you about stories 1, 2, and 4. So let's start with the first story. So this is a bird's eye view of our approach. I'm just reminding you what we're going to do is to construct a temporal network that describes how the Th17 differentiation process is regulated, starting from like 1 hour after we induced the differentiation until like 3 days later. So this is a bird's eye view of our approach. We start by collecting a time course of gene expression data as we follow the cells as they differentiate from the naive state into Th17. Then, based on this data, we build a regulatory network model that describes how these expression changes are being regulated. And then, in order to understand the function of this network and to validate this network, we designed a set of experiments, of perturbation experiments, where we knock out selected genes from the network and measure their effects on gene expression. And then we can actually roll back and look at these experimental data and then try to build, to substantiate our model and try to maybe improve it a little bit and come up with very specific circuits that describe various aspects of TH17 differentiation. So I'll walk you through this process. So as I said, we measure gene expression data as we follow the cells half an hour after the differentiation, up to 3 days. And as control, we use TH0 cells, which are basically naive T cells that are being activated, but not treated with any additional cytokines. And when we compare these two time courses, we find about 1,300 genes that are differentially expressed, and they exhibit very diverse spectrum of temporal profiles. Now, without going into details, we found these differentially expressed genes using a consensus of several statistical methods for differential expression, and this is just one of them. So let's say we want to compare this time course to that time course. So instead of comparing them directly, what we do is to fit a mathematical model, to fit a curve to each one of these time courses, and then what we do is actually, instead of comparing the actual data, we're comparing the fitted curves. And this actually has several advantages. So first of all, it's very robust to transient fluctuations in the data. And it's actually, if you want to impute some data, it can be a good tool. We actually also use this continuous representation of the time course in the later class. I'm going to talk about just in 2 slides. So we have our differentially expressed genes, and now what we want to do is to understand how are these genes regulated. In order to identify the regulators that drive these temporal profiles, we cross our time course data with a database of potential protein DNA interactions. So it can come from, it's a big database that comes from ChIP-seq experiments, from motif analysis, from interactions that are inferred based on correlations in mRNA levels, and even IPA. I know that there is some IPA talk here later, so we also use IPA here. So, and just to illustrate how this process works, so just imagine we have ChIP-seq data for our gamma T that significantly overlap with a cluster of late-induced genes. Now, if this overlap is significant enough, we're just going to connect, in the network, we're going to connect our gamma T to its binding targets that are also a member of this late-induced cluster. So far, it's pretty straightforward, but the question is, when do these interactions actually occur? So what we want to do is basically place a timestamp on every interaction, and we do that by looking only at time points where the target gene that something happens to the target gene. It's either induced or repressed or differentially expressed. But we also want to require sufficient amounts of the regulated protein. Now, since we only have gene expression profiles in our hands, we need at least some computational way to at least crudely estimate when do we have sufficient amounts of protein, and we do this by combining these different models here. So one model is a continuous representation of the mRNA profiles like I showed you before. The second one is prediction of protein half-life, and the third model is an ODE model. Basically take these two and combine them together to predict protein abundance. And just as an example, the model predicted that our gamma T reaches sufficient levels of protein after about 20 hours, which exactly coincides with the time of induction of its known target genes like IL-17 and other TH17 signature molecules. And we can see that this prediction is actually very nicely validated with the FACS analysis. So now with these timestamps, using these estimates, we can now infer for each time point which are the regulatory interactions that are actually taking place during that time point. And altogether we are getting this smoothly evolving model of 18 networks, one per each time point, that describe the regulatory landscape, I guess, throughout the differentiation process. Now, looking at this model, we were actually encouraged because it recapitulates a lot of the known biology on TH17 differentiation, which was a good sanity check. So, for example, the early networks contain known early induced factors like IRF1 and STAT1 that are predicted to cross-regulate each other and together control the induction of early induced genes. And then another example is the transition, the master regulator, our gamma T, is only active at the later stage of the differentiation and only present at the late stage networks. And this is in contrast to the pioneer factors like STAT1 and IRF4, like STAT3, IRF4, and BATF that are actually present throughout the differentiation process. So these are all nice examples, but what was more interesting in this network, and more important, I guess, is the fact that it's over 50% of the regulators in this network are completely new to TH17 biology. So that was a very good property that allowed us to address the next question that we have in mind, the key question, which is which perturbations we can do in order to disturb the differentiation process. So we treat it as a classification task, okay? The classification should be will or will not have effect on TH17 differentiation. And the features that we have are, we have these two sets of features. One describes the temporal activity of a regulator node in the network, and the second one characterizes the expression of the encoding gene. And both of these feature sets are actually highly predictive for genes that are already known to have an effect on TH17 differentiation. So we combined these two together, we created a red list of genes, and then we walked our way down the list and basically selected the top 40 genes in that list for follow-up analysis. And importantly, over 50% of these nodes that we selected, of the genes that we selected, are new to the TH17 field. So just to recap what we had so far, we started by collecting this time course of gene expression data. We inferred this preliminary model of regulatory interactions during the time course, and then we used that in order to understand the function of this network. And to validate this network, we designed this set of perturbation experiments, which we want to do now. So in these follow-up experiments, what we do is we knock down each one of our selected genes, and there we use this silicon nanowire technique. That's the only thing that worked in primary T cells. And then for which of these knockdowns, we measure the effect on the expression of a set of signature genes, about 300 genes in this cohort. And then we identify the cases where the knockdown had a significant effect on the expression of genes, either upregulated or downregulated. So when we collapse all of these significant effects together, we get this blob here, where a node A is connected to a node B if the perturbation of A had an effect on the expression of B. And the edge would be red if the effect is positive, meaning that B was downregulated, and blue if the effect was negative, meaning that B was upregulated. Yes?
So just expand a bit on how the knockdown is performed. So this is siRNA.
siRNA, yes. Okay, so is that the question? Yeah. All right, so now we can scratch our heads and look at this network and ask ourselves whether there is any structure in this network, whether we can find anything interesting in this network. So we start our quest for structure by, so of course there is a structure, otherwise I will show it to you. So we start our quest for structure by focusing on a small set of TH17 signature. We then highlight all of the genes that are positively connected to our TH17 signature genes, so like a TH17 positive module. And what we find is that the TH17 positive module is negatively connected to signature genes from other CD4 positive T cell subsets, and specifically to FOXP3, which is the master regulator of the regulatory T cells. Now, when we look at the TH17 negative module, we see the mirror image with positive interactions with the competing T cell lineages. And finally, when we look inside the modules, we can see that both of these modules are tightly intra-connected by positive interactions and interconnected by negative interactions. So abstracting away the individual edges, what we find here is that we have this system of tension between two modalities. We have the TH17 positive module, we have the TH17 negative module that cross-regulate, that antagonize each other, but promote themselves, and have an opposite effect on the balance between TH17 cells and other competing T cell lineages, and specifically between TH17 cells and regulatory T cells. Now, when I looked at the image, it actually took me back to some work that I did in my PhD, and it makes us think that maybe what we see here might be more of a general principle that we will expect to see in this kind of this type of biological network, this like bistability kind of structure. So this work was done in yeast, where we actually investigated a network which is exactly the same type of network, but much, much larger. It has about 24,000 edges, and it's based on knockouts of over 200 genes. And what we find is that over 80% of this huge network can be completely explained by this very simple model of red versus blue that on these two modules. And just by observing this very simple structure in the network, we can actually design very simple but very effective prediction algorithms for predicting the effect of knockouts. And this algorithm actually did much better than previous methods that were much more complicated algorithmically, just because it puts the finger on that very simple structure that we have in the data. And there is actually, in this paper, we also looked at the cases that deviate from this linear behavior, from this two-set behavior. And that was actually interesting cases for us to look at. I won't go into details here. I will just say that it had to do with compensation mechanism of the cell. This is just another example from another paper from Roy Kishoni lab that showed similar kind of structure in the metabolic network also in this. So going back to Th17 cells, so this model that we have, so it highlights some of the logic behind the regulation of Th17 differentiation, but it also highlights 12 genes that we identified that were complete, so that we see their effect on Th17 differentiation, but they were completely new to the Th17 field. So obviously these genes were interesting for us to look at more closely, which leads me to the last part of this project, where we actually go back and do another set of experiments in order to really investigate these new genes and really substantiate their role as real effectors on Th17 differentiation. And we use a slew of experimental methods like RNA sequencing, ChIP-seq, and knockout mice protein level analysis, and other types of assays. And I'm going to show you some of these results in the next few slides. So, so this is just some highlights from this specific study. And basically what we can see that by overlaying this new experimental data that we have on the edges from our regulatory network, we can actually identify and further really drill down into a number of novel circuits and then we can actually also go back and see whether these specific circuits give us some hypothesis for this higher level functional question that we started up with. And this is just one example. So this is a circuit that is centered around the chromatin regulator MIEP, which is a member of the Th17 positive module. So MIEP is predicted to have its effect through positive feedbacks with two of the main transcription factors in the Th17 network, Aura-gamma T and VATF, if I can see from here. And it also has a negative edge, a suppressing effect on the expression of FOXP3. And what we can see is that if you look at the knockout mice of MIEP, we can actually, it supports this model where we see a marked decrease in the differentiation of Th17 cells, but we do see a nice increase in the abundance of FOXP3. This is just one quick example. I want to give you a bit more in-depth example in the next study that I want to tell you about. And in this next study, we look more closely into one of the aspects of this network, and this is the basically number 2 on my list, which is a signaling regulatory model of signaling downstream of IL-23. So just to give you, just to remind you some of the things we talked about in the beginning, it is already known that signaling to IL-23 receptor is crucial for the stability of Th17 cells. So you can view the IL-23 receptor as a member of the red module that we saw in previous slides. So this is already known, and this is just some data to show you that IL-23 signaling to IL-23 is maybe not so important for the early steps of the differentiation, but it is important for the stability. Because if you are comparing these expression profiles between wild type and IL-23 receptor knockout, we can see that after 2 days, which is here, the cells look more or less the same. But then if you wait another day, you can see that you're losing the Th17 phenotype and you're starting to get some gain of the cytokines and transcription factors from the competing T cell lineages. So all of these things is known. Actually, IL-23 is one of the poster childs of genome-wide association studies, a very, very famous paper that came out in Science in 2006, 2005, that actually links IL-23 receptor and other members of the IL-23 receptor pathway to IBD. So it's a very well-established molecule, but what is not known is what is the mechanism, or how does the IL-23 receptor signal actually propagate from the cell surface to the nucleus, and actually affect the transcription of genes. So that's a question that we have in our heads. And we address this question by extending upon the framework that we saw on previous slides. So we start by finding genes that differentially expressed in IL-23 receptor knockout in the time course again. Then we identify the transcription factors that are dysregulated in this process using the same method as I showed you before. And now what we want to do is to find the most probable pathways of protein-protein interaction that will connect IL-23 receptor and some of the associated upstream molecules to these transcription factors. So how do we do that? Basically, if we, on our first stage, we used a large database of protein DNA or potential protein DNA interaction to actually infer these interactions here. So now we're going to use a second database of protein-protein interaction or potential protein-protein interaction to actually address the other layer of this model. So this is a large database, about 80,000 interactions collected from the literature. And every interaction in this model, in this database, is associated with a confidence score, some number between 0 and 1 that says how much we believe that this interaction is actually a true measurement, and this is just being learned automatically based on the experimental evidence behind that interaction. And now what we want to do is to extract from this large database the best network of protein-protein interaction that will connect IL-23 receptor and the associated molecules to our transcription factor. So now the question is, what is the definition of best? You can think about it in two ways. One is to say, I want IL-23 receptor to be close, to be near my transcription factors. So basically, I want the path to be short. So the algorithmic way to approach it is basically finding shortest path. So the advantage is that it is finding the most efficient way to reach every transcription factor individually. The disadvantage is that we ignore possible dependency between transcription factors because they actually solve the problem separately for each one of them. The second problem is the second approach is to say, let's look at all of them together and find the overall most high confidence network that connects IL-23 receptor to all of the transcription factors together. So in computer science terms, it's called the Steiner tree problem, which unlike shortest path is an NP complete, but there is some good algorithms to solve it. So it's good because it takes into account the dependency between the transcription factors, but the problem is that it's very sensitive to false positives in the data. And it can also lead to very long paths in some cases. There is actually, you can show just by mathematical ways that there is actually a very nice trade-off between these two approaches. You can do very well here and very bad here and vice versa. So what we actually want to do is try to bridge the gap between these two approaches. And this is exactly what we did. So we developed this algorithm that can actually find the sweet spot, the best combination, the best compromise between these two objective functions. And this is actually, we can actually even prove it mathematically that it is guaranteed to give us the best combination between these two, or approximately at least the best combination between these two objectives. So we implement this algorithm in a tool called the software tool called ANAT, which you use in various other systems, but now we also want to use ANAT in IL-23. So we applied ANAT on the IL-23 expression network, and on the IL-23 network, this is what we're getting here. So in blue are the upstream nodes, the IL-23 receptor and the associated molecules there. In green are the downstream transcription factors, and the rest of the nodes are basically predicted by ANAT to act as mediators in this process. Now again, we can scratch our heads and look at this network and say, what can I gain from this ball of yarn? So one of the things that it can give us ranking. We can actually basically rank the nodes in this network based on how central they are in funneling the signal from IL-23 receptor. But one thing that we must be taught is that we need to be very careful when doing this type of analysis, because in this kind of large database, you can connect anything to completely meaningless pathways. So there is a lot of steps that we need to do in order to go from the theory of, from graph theory to actual application in these kind of cases. And one of the things is to assign statistical significance and basically say for each one of these mediators how much we'd expect to see just with random input. So we actually filter out all of these spurious predictions from the network and only remain with the ones that we trust. So the most highly ranked gene in this network is the protein kinase SGK1. So SGK1 actually already caught our interest based only on the gene expression data. We actually see that it's very strongly regulated by IL-23. So basically it is very strongly down-regulated when we knock out IL-23, and it is also very nicely overexpressed in the TH17 differentiation. So it was already on our radar. So what do we know about SGK1? So it's a pleiotropic kinase. It plays an important role in stress response, in cell cycle, in apoptosis. And another fact that I'm going to return to in a couple of slides, that it also regulates sodium transport and homeostasis in the cell. Now, SGK1 was not previously implicated in TH17 biology, but given all this mounting body of evidence, we decided to look more closely into it. So using the SGK1 knockout mice, it actually became clear that it is essential for TH17 maintenance and for TH17-mediated autoimmunity. And specifically, we see that in vitro, TH17 signature genes are completely repressed in SGK1 knockout. But more importantly, in vivo, we see that SGK1 deficient mice are resistant to EAE, which is a mouse model of multiple sclerosis, specifically due to defects in TH17 maintenance. So now the question was, we found like one link in the path underneath of the IL-23 receptor, or one node. The question is, what is the mechanism? What is downstream of SGK1? So to address this, we basically repeated the same analysis as before. This time we are looking at SGK1. So we find the genes are differentially expressed under SGK1, find dysregulated transcription factors, and then do the protein-protein interaction analysis. And what we find in the end is this, we kind of converge to this model. So this analysis actually highlights another factor, which is the transcription factor FOXO1, as an important downstream target of SGK1. And together we get this logic here where FOXO1 normally acts as negative regulator of TH17 differentiation. For example, it binds directly to the IL-23 promoter, and then SGK1 can phosphorylate FOXO1 and then basically deactivate its transcriptional activity. So in a sense, what you can see here is that we have this very indirect positive feedback between IL-23 receptor and SGK1, and this positive feedback is a characteristic of the stable state of TH17 cells. And when we actually disturb this feedback, either by knocking out SGK1 or by messing with IL-23, we can actually lose the TH17 feedback. I told you just 2 slides ago that one of the known goals of SGK1, one of the known goals in the literature is as regulator of sodium channels, and it's actually also induced by sodium. So the natural question to ask was, what is the relationship between sodium and TH17 differentiation? Or what will happen if you just take naive T cells and culture them with elevated levels of salt? And what happens is that these cells actually differentiate into TH17 cells. And this happens both, and we see evidence for that both in vivo and in vitro. And we can actually see that also in human T cells. That's an accompanying paper that came out from David Huffler's lab. And what was another study we did in vivo was to actually take mice and feed them with, compare mice with high salt diet to mice with regular diet. And you can see that the mice with high salt diet can actually develop much more severe autoimmunity. Yeah, not oral enzymes. Yeah. So basically, we are very careful about. I mean, basically, what it gives us is it gives us some sort of molecular level mechanism that explains the effects of this very high level environmental factors, such as high salt diet on autoimmunity. And this is just one hypothesis that one way by which this signal is mitigated is through TH17 phenotype, and specifically through the activity of SGK1, because we see that we actually knock out SGK1, we lose the effect of sodium. But of course, we need to be very careful about making statements about how comprehensive this model is, because changing the concentrations of salt can have a lot of effects on the behavior of the cell. But this is just a good starting point to start looking at this very important effect that we see. And I can tell you that we already have ongoing, at least in the planning stage, advanced planning stage, large experiments in human cohorts. We're trying to actually see the link between diet and autoimmunity and see whether we can drill down and understand better the effects that we see here. If there was any question? Okay. So far we talked about how can we use the variation of cells over time to actually do TH17 differentiation in order to discern the mechanism behind the regulation of these cells. So obviously these cells change over time. The question is, what about variation between individual cells in a population of TH17 cells, and whether it is even interesting in the context of TH17 cells? So the answer is, of course, yes, otherwise I wouldn't ask the question, but this is actually, it is, okay, I'll talk about this in a second. It's already been known that the term TH17 cells, this is something that comes up in the literature maybe in the last 2 years, that the term TH17 cells actually covers a very wide range of subtypes of cells. And this is a view that I like. This figure came out in this review paper from John O'Shea a couple of years ago that makes a very, I guess, bold claim that if you open the immunology textbook and you see the canonical T cell types that you have TH1, TH2, TH17, but what actually happens when you look in vivo is all of these like creatures that lie in the middle between these canonical states, all of these double producers, all these cells, they're actually somewhere on the spectrum between these canonical states. And why is it important? Specifically, why is it important in our context? So it's important because we already know that these specific subtypes of TH17 cells can have very different pathogenic effects. This is a paper that we had a couple of years ago. We showed that under some conditions we can generate TH17 cells that can be very pathogenic in an adoptive transfer model, and others that can have no effect at all. And this is something that we can see also in vivo. In some cases we can see that the TH17 cells are very pathogenic, and in some disease models they even produce immunosuppressive cytokines that are maybe considered to actually act as the repressive for the immune response. So what we'd actually like to do is to get a global understanding, an unbiased understanding of the subpopulation, the subset of TH17 cells, and specifically to identify this pathogenic subset of TH17 cells. And by identifying this pathogenic subset of T cells, wherever they lie on this spectrum, we're able to actually develop much more efficient therapies against various autoimmunities that we are interested in. So how do we do that? How can we, in an unbiased way, identify a subpopulation of TH17 cells, which is supposedly a homogeneous population of cells? So of course the answer is using single-cell RNA sequencing. And that leads me to the fourth and last project that I want to tell you about, which is how can we use single-cell gene expression data to actually infer these subpopulations. Now, I should say that this is an unpublished work, and this is kind of a still work in progress. So I would be happy to get any feedback from you on whatever I show you. This is really still in the making. So that's how this is how our data looks like. This is a huge matrix. We have about, let's say, about 6,000 rows here. Each row is a gene, and about 1,000 columns here. Each column is a cell. And we have we collected cells both in vivo and in vitro. In vivo, it's in an EAE model at the peak of the disease, about 2 weeks after induction. And we have cells both from the central nervous system and from the draining lymph node. And from the in vitro condition, we have basically 2 cytokine treatments, one that leads to the non-pathogenic T cells, and the other one, that TH17, the other one that leads to the pathogenic TH17. So IL-26 plus TGF-beta1, this is the non-pathogenic, and IL-1 beta, IL-6, IL-23, which is the pathogenic TH17 cells. And again, the reason I call them pathogenic and non-pathogenic is just based on our results from adoptive transfer experiments. So now the question is, how can we tease out the information from this matrix in order to say something about subpopulation of T17 cells? So before we actually start addressing this question, there is a lot of computational work to be done. A lot of things that we need to think of. It's because this whole field of single-cell transcriptomics is a very new field. And a very exciting time, I guess, to be a computational biologist because it's a very new field, very important field, but a lot of open computational questions. And the reason this slide is so dense is because there is so much to do, I guess. And I just highlight here some of the interesting tasks. And today I'm going to, in terms of the computational tasks that I'm going to show you, I'm going to talk about two of them. One is the more mundane but very important task of pre-processing the data, which can be extremely noisy, extremely problematic. And the second one is how can we infer subpopulation and actually understand the subpopulations. So let's talk for a second about pre-processing. I just want to make a point here that this is something that I've seen in all of the single-cell data sets that I've been working with so far, is that if you just, let's say we're doing RNA sequencing, let's say for like 200 cells, we're taking these 200 cells, and we do, let's say, clustering, just as is, without touching the data, the cluster that we get, or the principal components that we get, will reflect the biases in the, will almost certainly reflect the biases in the library efficiency, rather than anything that has to do with biology. So that means that we really need to make sure that we are filtering out all of these biases from the data before we do anything with it. And this is just a list of these measurements for library efficiency, like 3 to 5 prime bias, like complexity, like the number of transits that we can actually identify, number of reads, etc. So this is what I told you about. If you just take these, if you just take the data as is, do principal component analysis, and correlate it with these parameters, we can see a huge amount of correlation. So we developed this pre-processing pipeline that actually composed of 2 steps. One is filtering cells that are not doing well in terms of these parameters. And the second one is actually using a method that was previously used for removing GC content biases from RNA-seq data to actually, in a quantitative way, remove these biases, basically shave them off the matrix of data as well. And we see that after we do these steps, we can actually lose these correlation between our principal components and these different covariates for the data. Another thing that we need to take into account is the very high level of false negatives in single-cell data. And in some of the single-cell papers that come out now, you can see that in some cells you have like 100 transcripts, 1,000 transcripts. Of course, this is a fraction of the transcripts that are actually there in the cell. So basically, the fact that I do not see a gene in a certain cell doesn't necessarily mean that it's not there. It can be a false negative. So we need some way to actually take this into account. So what we do is we build this characteristic curve of false negative rates for every individual cells, where given the expression level of a gene in the population, in the population, we can actually predict what is the probability for false negative. So basically, all of the subsequent analysis that I will show you is weighted by these false negative estimates. So now let's leave the pre-processing and go to the interesting part. When we look at the gene expression profiles of the different genes across the different cells, and we look at the histogram of every genes, basically we get 2 types of genes. We have genes that are considerably expressed across all the cells, like RRT, like BATF, IRF4, etc. Not surprising, and housekeeping genes. But then we have genes that are bimodally expressed. Some of the cells express them, and some don't. So basically, these genes are characteristic of subpopulations of cells. And when we look at these genes, we see some interesting cases here. We see IL-10, we see IL-17, we see even FOXP3. There might be some important information here. So what we actually need to know is how do these genes actually co-vary with each other. And we look how they co-vary with each other, we get this very nice image, as you can see here. Basically what we see is that we have these 2 modules. So basically every column here is a gene, and what we see here is the correlation of each gene with a small set of these signature genes that we have here. And we see that the genes in the genome are actually partitioned into 2 sets. The ones that positively correlate with IL-17 and negatively correlate with IL-10, or vice versa. So we basically see this antagonistic structure between IL-10 on one hand and IL-17 on the other hand, or the non-pathogenic phenotype on one hand and the pathogenic phenotype on the other hand. And when we look at the principal component analysis of these Th17 cells, we can actually see exactly the same picture, where we can have this very nice separation into T cells that have high levels of IL-17 and ones that do not have high levels of IL-17. And as you go to the right in this principal component map, the cells on the right side are are characterized by a very pathogenic signature, where the ones on the left are actually characterized by production of IL-10 and more non-pathogenic kind of signatures. Basically, we see this very nice spectrum of behavior between non-pathogenic Th17 cells on the left to pathogenic Th17 cells on the right. Now the question is, what is the meaning of this spectrum of behavior that we see in vitro? What is the meaning of this spectrum in vivo? So I won't go into I guess I have 15 minutes, so I can tell you why. So yeah, so basically what we see here is the spectrum of behavior of Th17 cells from a non-pathogenic phenotype to a very pathogenic phenotype. And now when we look in vivo, so I remind you, we profile T cells from the lymph node and from the brain. And so when we look at the principal component analysis of these in vivo cells, we see a very nice separation between the brain cells and the one from the lymph node. Which was nice to see, that we can actually separate them. But more importantly, what we see here, that once again we have this kind of this, if we try to actually characterize these cells using additional information, we can see that we have this, as you go here from the left to the right, we can see this spectrum of behavior again, as the cells from the left are more characterized by a naive T cell, like a phenotype, and self-renewal phenotypes, like Wnt signaling and TCF7 expression. Whereas the cells on the right are more characterized by terminal effector kind of profiles or memory profiles. So basically what we see, the spectrum of behavior that we see here in vivo agrees with that, this image here, that it's kind of a speculative image that came out in a review paper about a year ago, that basically claims that we have this gradient of ability, of capacity for self-renewal that actually decreases as you go from the precursor Th17 cells, and actually decreases as you go basically from these precursor cells to the cells that actually go to the site of inflammation. And this is exactly what we see here. Basically, these are the precursor cells, and as you go from the lymph node to the brain, to the site of inflammation, you basically lose the capacity for self-renewal and acquire all of these terminal effector phenotypes, and also like a Th1-like phenotype that characterize the pathogenic activity of Th17 cells. So now the question is, what is the relation between this spectrum that we see here in vivo, that basically characterizes the activity of Th17 cells, and how do they move from the lymph node to the brain? How does this spectrum here reflected in our in vitro data? And we can actually see that it's reflected very well. If we're taking this axis here and actually project it into our PCA plot in vitro, this is what we're getting. So basically it means that the cells that are here on the right side, which are the more pathogenic cells, they are also characteristic by being on the right side here, basically being a characteristic of the terminal effector cells that are actually at the site of inflammation, that have the Th1 phenotype, and actually induce the inflammation in EAE. So this gives us another support for the spectrum of behavior that we see here, basically from this non-pathogenic Th17 cells to the pathogenic Th17 cells, and we can interpret what is the meaning of this thing when we look at the in vivo model. So that's good. Now we can have this. We have this spectrum of cells, and now we can actually go back and make use of the fact that we have genome-wide gene expression data, and see if we can actually gain anything new by looking at this map. So what we basically want to do is try to compare the cells on the right to the cells on the left, and ask what is the difference between them. And by looking at the differences between them, see if we can actually find hypotheses for new regulators that are specifically important for regulating the pathogenicity of Th17 cells. And that's exactly what we did. So basically by comparing these two sets of cells, we could identify a number of novel regulators. We actually tested 7 of them using knockout mice, and for 4 of these cases, we could actually find very, very strong phenotype on Th17 function. And this is just one example. It's the G-coupled protein receptor 65. is a glycosphingolipid receptor that we can see a very specific effect on pathogenic Th17 cells. So this is just an example for an EAE model where we see that the GPR65 knockout mice do not develop the disease at all. And this is just in vitro analysis, we can see that the effect of the GPR65 knockout is the most pronounced in the pathogenic subset of Th17 cells. So that gives us actually that completes some previous hints that came out from the literature, some of them based on genetic data, and give us some potential mechanism for why and how GPR65 is important for regulating Th17 activity. So, yes. Yes. Oh, no, it is it. Yeah, sorry. Yeah, so just to summarize. So I showed you a bunch of methods that we have. Basically, we can see how we can, using time constraint expression data, we can construct a temporal model of the wiring networks and how we use it for Th17 differentiation. We extended this method to actually also build signaling layer on top of the regulatory networks, and we applied it on the alpha-1 receptor. And then we have this bunch of computational tools that we developed for analysis of single cell data. In terms of mechanism, this analysis, we actually found a number of new hypotheses. One is this bistable model of Th17 differentiation that I showed you, this new signaling pathway downstream of IL-23, and the sodium effect on Th17 differentiation, and the different subpopulation of Th17 cells. In terms of the function, these kind of mechanistic things that we found gave us some hypotheses for the functional type question that we started up with about how the balance is maintained between regulatory T cells and Th17 cells, for example, through MIEP, or about the stability of Th17 cells, for example, through this SGK1 FOXO1 pathway, or about what makes these cells pathogenic. So, for example, here we had this GPR65 and other results that we have from the single cell data. And about the application, so I can tell you that we already have some encouraging results in vitro for small molecules against SGK1, and the study that I didn't tell you about today about small molecules that target our gamma T, which we just published. So this just to acknowledge the people that were involved. So all of these studies were done at the Broad Institute at Harvard Medical School. My previous postdoc advisor, Aviv Regev at the Broad, Vijay Kutru at Harvard, Chuan Hu and Teresa Talamer are postdocs at Vijay's lab that worked on the SGK1 project. Ramkun Park is our collaborators at chemistry and physics at Harvard, and his students, Yael Moglumi and Alex Shelek, were involved in the single cell experiment and project and the first project of the temporal regulatory network of Th17 differentiation. And with that, I will conclude. Thank you very much. So the question was if we had experimental data to support or to use for the protein abundance model in the first network model that I showed. So we did, there was one experiment we did with our gamma T that I showed, and basically measured the abundance in several time points. And what we see using this FACS analysis actually supports what the model predicts. So that's the one thing that we had. We didn't have additional protein level information there. I can only say that we tried to be very conservative about using this model, and in a sense, try to exclude things, then try to go more strongly on the false negative than on the false positive, I guess. there in terms of presence of protein. So basically we're just trying to be more conservative because it is very hard to make these kind of predictions. No, it doesn't. Yeah, so the question is whether the model incorporates post-transcriptional, post-transcriptional or probably also post-translational. No, it doesn't. And there is definitely, it can definitely be taken into account. There are additional types of data that we probably need to have in order to do that, which we didn't have in this case. But I can tell you that this is definitely something that people are thinking about these days at the broad. Yeah. You mean in the single cell?
The one where you have the TH1 subtype versus the more IL-17
Yeah.
subtype of the TH17.
So
some of those are not producing IL-17. We didn't select it on IL-17 production.
No, so in this case, what we did was basically use TGF-beta1 plus IL-6. Normally, TGF-beta1 plus IL-6 gives us about 50-60% in mice of IL-17 positive cells. But the interesting thing is that all of these cells, all of them, the vast majority of them express our gamma T, and they express IL-17F, which means that they have a lot of TH17S in them, and it is interesting to look at them as a population. We also have data that we use GFP positive, IL-17A, GFP positive for IL-17A, but actually this data is much more informative, and it really gives us this spectrum that we are interested in seeing.
The reason why I'm asking, I was wondering if these cells, if you included, and maybe you've already done this, but if you were to look at the non-polarizing situation and just let the cells develop a given antigen, is there even such a thing as an IL-17 value to such a cell?
No. IL-17 is not expressed at all in TH0 condition. Yeah, I think
We assume that because cells produce IL-17, that is a TH17 cell that's similar to TH1 and TH2, where the transcription factor, the protein production, the surface expression all kind of come in hand. And you're clearly showing that this does not all go hand in hand with TH17 cells. But what I'm trying to ask is, do you think that it would even define subset, or because a lot of your data kind of implies that it develops into other things at some point? So
Yeah. So I'm not sure I followed the question, but I will just say that we definitely see different subsets of TH17 cells, and the fact that we see different subsets is very tightly linked to the plasticity of these cells. So, for example, the plasticity or the change of phenotype for the TH1 phenotype. This is something that we definitely see, especially in vivo. We see that the cells start producing interferon gamma and other like IL-18 and other TH1 kind of genes. We see it less in vitro, but much more profoundly in vivo. In terms of what you asked about, I don't know if that answers the question, but just one thing about just activating naive T cells in the plate. If you do that, you will not see, at least from the data that I've seen so far, we do not see any IL-17 production at all there. What we might see a little bit is interferon gamma. So in a sense, maybe like the TH1 kind of wins. Maybe there is some autocrine signaling there that.
Do you think your red dots will cluster better with effector TH1 cells than they cluster with the green dots?
No. No, because these cells have a lot of, basically what we did was to take these cells and try to characterize a signature of TH1, a signature of TH17, etc. And of course, we see that throughout, this TH17 signature is the most pronounced. So they do look like TH17 cells. They do express our gamma T, which is something that is a very strong hint. Basically tells us that this is like a pan-TH17 factor. And then when we go down, so we can see basically, everywhere we look, we see our gamma T, as you can see here. But then we have these subtypes of cells that are characterized by the genes in the upper part of this map. And one of them is IL-17A.
One last quick question.
I'm actually following up on this discussion. I was wondering, in this, those two subpopulations, the IL-10, the more IL-10 leaning subset of B17, how are they in expression of regulatory factors and FOXP3? How close are they to Tregs?
So that's a good question. We don't see a strong Treg signature there. It might be a result of false negatives that we just can't, because FOXP3, for example, is kind of lowly expressed, kind of hard to catch. But we do see a nice module of immunosuppressive cytokines. So we see IL-9 and IL-24 and IL-10. So that, so as a whole, it makes sense to us.
Because there are tons of works, and we actually saw a couple of them this week. That this shift of IL-17 and Tregs and FOXP3 versus overall gamma T is, they're very mutually regulating each other. And it's like if you turn one off, then the other one also switches on.
Yeah.
It might be that you're seeing here a shift towards a Treg population, not so much as a subset of T cells.
It might be, but let's say that we cannot say that based on the data that we have in our hands.